Hello, everybody. I'm Robert Boyd from the University of Ottawa and the University of Rochester. And I want to welcome you to this, the second day of our Schawlow-Townes Symposium for 2020. It is really sad that we cannot get together in person for an event of this sort, because the interpersonal connections can be very, very useful for the development of science. But we have to live with the rules as they are handed down to us. Before I want to go on, I want to thank the two sponsors, the primary sponsors for this event. It's the University of Ottawa and the National Research Council of Canada. So our thanks to both of them. And let me move now directly to the first talk. Our speaker is Jochen Guck from the Max Planck Institute for the Science of Light in Erlangen, and he's also a professor at the FAU Erlangen-Nuremberg University. Jochen Guck studied physics in Würzburg and then went on to receive his doctorate from the University of Texas at Austin, USA. I just a minute ago complimented him on not having developed a Texas accent. He later worked at the University of Leipzig, the University of Cambridge, and at the Technical University of Dresden. And since 2018, he has been a director and a professor in Erlangen in Germany. So Professor Guck, I welcome you to this event.
Well, thank you very much. I will share my screen. Okay, so hello everyone, my name is Jochen Guck, as you just heard. Thanks for tuning in. I would like to start by thanking Robert and Paul for inviting me. And I consider this a great honor to be a speaker at this symposium. It is rather unfortunate that the meeting doesn't happen in person, that I cannot be there in person, because I would have really liked to finally visit Ottawa and to get to know you better. But I think this is what it is, and we'll have to make up for that at some later time. I want to start with two general announcements, if you want. One is, I'm sure that you're aware that Arthur Ashkin died a few weeks ago. And even though I've never met him, he's my personal hero. I think it's fair to say that without him, I would not have had the career that I had. And in particular, his pioneering work on optical trapping very much shaped the way I took in my career. So I would like to dedicate my presentation today to his memory. The second general announcement, I guess, that I want to make is that, well, you see the title here of my talk, it's Feeling with Light. I will be talking about optics and a bit of photonics, I guess, but this will not be fast nor extreme, and certainly not quantum compared to anything that you guys are doing. So I hope that you still enjoy my presentation, which uses rather basic optics, but has some great insights, or how we use light and optics to get great insight into very fundamental biological processes that also have the potential to ultimately impact clinical medicine. With that, of course, light has had a large impact in the evolution of the history of biology. Everyone knows that from the time of Leeuwenhoek and Hooke, who invented the microscope, figured out that there was something like cells in the first place, all the way to today's science and rather late Nobel Prizes here on the use of light for super resolution microscopy. Light has been central in the discovery of what life is and how cells work, etc. And in particular, pushing resolution down to almost molecular level, I think is very much in line with the state of biology, which is essentially molecular today, right? Ever since we figured out the central dogma of molecular biology, DNA mix RNA mix proteins, we know the human genome and that of many other species. We can quantify how much RNA is in a cell, of which kind, and proteins, etc. It certainly makes sense to also have an imaging technique that then can look at where what is. But the question is that sufficient? Is it sufficient to know what molecule is where and when in a cell? Right? If I, as a physicist, if I look down a microscope and look at a cell, I see more than just a bag full of molecules that are reacting in chemical reactions with each other. That there are clearly emergent phenomena at different scales. Right? One particularly hot topic at the moment is the phenomenon of liquid-liquid phase separation that generates organelles inside of cells that have function. But then also even the entire cell is more than just a collection of molecules. It clearly is a physical object that has material properties, has optical properties, has mechanical properties. And I was always fascinated by the question whether these cell-scale physical properties have any meaning in biology. And that's what we're after. And I think the divide between, or the transition from molecules to entire cells and cell physical phenomena can probably be illustrated very well by trying to answer the question of why a plane flies. And if you put yourself in the shoes of a molecular biologist that probably would sit down in the cockpit and say, I have all these beautiful knobs and switches, and clearly if I switch them, I get some response, and that's if I know how to do that, and this is why that plane flies. But as a physicist, if you take a few steps back and look at the plane from the outside, you notice that it has a particular shape, right? It has wings. The wings are more curved at the top than at the bottom. And so if there's enough forward motion, then the pressure difference will be sufficient that you generate lift that can balance gravity. And that's why the plane flies, right? So both questions, both answers to that question are valid, but neither one of them alone would probably give you a full picture. So I think it's important to not just look at the molecular level, probably level control of what the cell is doing, but you also have to consider cell scale physical properties in order to fully understand what cells are up to, how they function, how biology works. So I think what we are witnessing at the moment could be the next revolution in biology, in that I think we are about to experience a paradigm change from considering the chemistry of molecules to thinking more about emergent physical phenomena at cellular scales. And as any revolution, I think that requires the right toolkit. And I very much like the statement by Sidney Brenner, who said, developments in science depend on new techniques, new discoveries, and new ideas, probably in that order. So we need the techniques in order to move science forward, to make new discoveries and new ideas. Now, Erlangen is the perfect backdrop for this kind of paradigm change, driving that paradigm change, because we have the Max Planck Institute for the Science of Light. And I think light will continue to play an important and pivotal role in pushing our discovery into what biology is. So this is a great backdrop to develop the tools that we need to do that. And then we have this new Max-Planck-Zentrum für Physik und Medizin, which is actually quite interesting experiment within this building. We'll have researchers from the Max Planck Society, but also the University in Erlangen and the medical hospital together, trying to transfer our insights from this kind of biophysics type research into medical application. In fact, we just broke ground for this building this week. So this is a very exciting place to transfer, to have the tools and then get new insights and transfer that all the way to clinical application. Now, with this prelude, what do we actually do, right? So I said already, we're interested in cell scale physical properties. So in particular, we're interested in cell mechanics. We're interested in how cells measure and respond to the mechanical properties of their environment, which you could call cell mechanosensing. And we're doing that in particular in the central nervous system. We're also interested in not just mechanics, but also optical properties of cells, and have quite nice, interesting stories to tell in that regard. And as I said, we developed the tools largely that we need to do our research. So I want to start with tool development, because that already introduces my start into my scientific career, and that was very strongly influenced, as I said, by Arthur Ashkin, because it involved an optical trap. And this is a very simple optical trap that I guess you're all familiar with, a dual beam laser trap. You take two non-focused counter-propagating laser beams, and you can trap things in the center, because the scattering force pushes particles to the middle where the forces balance, and the gradient forces hold the particle on axis. So this was actually the first optical trap that Ashkin built. It was almost 2 decades before optical tweezers appeared on the scene, where you have a single beam that's very strongly focused, and then you have only the gradient force essentially balancing the scattering force. So this is a different, distinct optical trap, which has certain advantages, but is by far not seen the same usage as optical tweezers. Now my task was to set up this trap and then to trap cells. And we wanted to use the scattering forces to compress cells. So in a sense, we wanted to build not optical tweezers, but optical squeezer. And I did that. I was an undergraduate in Austin, Texas at the time. And setting up this trap is relatively straightforward. If you have single mode optical fibers, or you only need to align them and you have your trap, put the cell in, and then increase the intensity. And I was quite surprised to see the following. So this is a movie. And you can see when you increase the intensity of the light that's going through the cell, it is not being squeezed, but it's being stretched. So exactly the opposite of what effect of what we were expecting. And at the time, well, okay, we started calling this then optical stretcher for obvious reasons. But the imminent question was then, well, why does it stretch? And I was in a biophysics lab, more polymer, soft condensed matter background in the group than anything related to lasers or optics. So I sat down with a bunch of papers, and again involving one from Arthur Ashkin, who had shown in '73, when you send a laser pulse through a free liquid surface, that the surface wasn't pushed in as you would expect if there was the scattering force pushing it in, but it was actually lifted upwards. The image that you see was from a later repeat of that experiment by another group. But so this gave me the idea, well, maybe it's this, so this is caused by the transfer of linear momentum from the light to the surface. And if you go through the math, this is actually relatively straightforward why that would happen then. So that gave me the idea that maybe because we're sending light into the cell from one side, it's kind of the surface is pulled in this direction. If you do that on the other side as well, that's why the cell is being stretched. And it turns out that this is actually the explanation for this phenomenon. And we've done a lot of work on trying to understand that in detail. And I think we have clearly understood of why that happens, can calculate the stress tensor, and that quantitatively even then matches the stresses that we see that are required to deform the cell in this particular way. So that's why the cell is being stretched and not squeezed. That's the tool. So what do we do with that? I want to give you a few examples of what you can do with this optical stretcher. One was a lot of fun, actually, as it involved our sense of vision, seeing. You know how an eye works in principle, right? There's a lens, and that projects an image from an outside object onto the back of the eyeball. And there's a thin layer called the retina. And the retina contains then the cells that perceive the light, transfer that into biochemical information that's then sent on to the brain for further processing by the optic nerve, etc. So if you zoom into the retina, which you see here on the right in a schematic, there's something very curious in that the photoreceptors, these blue cells on the right side, they're actually furthest away from where the light comes in, right? So the light has to go through all these hundreds of microns of potentially scattering neuronal tissue before it reaches the photoreceptors. That's a very strange way to build an optical detector, right? You would expect the photoreceptors are at the front, and yet they're on the other side. And that has puzzled a lot of people for a long time. Also, I like this quote by Dick Feynman in his lectures on physics, who said about this: so some of the features of the eye are wonderful and others are apparently stupid, right? So how come that the retina is inverted and yet we can see with a certain level of clarity? And so we were, at the time I was in Leipzig then, we were working with a group that was interested in these yellow cells that you see in the schematic as well. These are so-called Müller glial cells, and they span the entire thickness of the retina from where the light comes in all the way down to the photoreceptors. So we developed this idea that maybe these are waveguides, maybe these are optical fibers that help pass the light all the way through to the photoreceptors. So we did some measurements. We measured the width of these cells throughout their geometry, and we measured optical properties, refractive index, and then calculated that indeed, when you calculate the V parameter, that these cells should be able to support a few modes of propagating light. Okay, so that was promising, but how do you test that? If you enzymatically get these cells out of the retina, this is how they look like, right? So how do you test whether these cells are optical fibers? And that's where this optical stretcher came in handy, because if you then bring the cells in between these two single mode fiber ends that you see here and turn on the near-infrared trapping laser, you see how the cells align onto axis and actually being stretched straight, and that for the reasons that I told you earlier. And now you can move the two fibers into contact with this cell optical fiber, and well, if that's a cell, then it should propagate light, right? So when you send in visible light, and that comes out of the mode field diameter of the core of the fiber, gets into the cells, passes them to the other side, you should be able to detect that signal. Indeed you do. So as long as the cell is present, you do get a certain level of transmission. And when you turn off the infrared laser trap, cell falls out and the transmission stops. So this was a very direct proof that these cells can act like optical fibers. And so together with a couple of other things that we did to show that, this was then also published in PNAS. We made the cover, and so we could, because I think this struck a lot of interest, because people were, had been wondering about why this inversion was not a problem. And now we were saying, okay, there are living optical fibers in the retina that help to propagate the light to where it needs to go within the retina, bypassing all this other potentially scattering tissue. So that was nice. We did some more work in that direction, but I don't have time to go into that. I rather want to return to this optical stretcher and something else that we did with that trap. And that relates to cell mechanics. Because if you can deform the cell, then you can measure mechanical properties. It's actually quite nice to combine this trap with microfluidic delivery. Then you have cells flowing along this channel. They are automatically trapped because it's an optical trap. All you need to do is turn up the light and you get a deformation, right? We know the stress, and all we need to do is then monitor the deformation. And this is how that looks like for a stationary cell. You turn up the stress, keep it constant, drop it back down, turn it up again, etc. And then you must monitor the strain, the linear relative extension along the laser axis, which is what's plotted as a moving plot at the bottom there. So that kind of information contains the full viscoelastic information of the cell as a material object. And we've had a lot of fun with that. We looked at all kinds of different biological processes For example, there are cells that are going through the cell cycle. They change their mechanical properties. So depending on what stage they're in, they have different, in this case, viscosity. Blood cells, when they differentiate into different lineages, they show different viscoelastic properties. If you look at cancer cells are always softer than their healthy counterparts, or immune cells, when they become activated, they become stiffer or softer in some cases. So I guess what you can take away from this slide here is that any physiological or pathological process that involves the cytoskeleton, so an internal polymeric structure within the cell that is involved in all these processes, so migration, cell cycle, etc. It also controls the mechanical properties. So whenever any process changes the cytoskeleton, you can feel for that. So you can take a cell, squeeze it, and figure out if the cell is changing its function because of that. A very generic way to test for changes in function in cells. And because it's so generic and has the potential to be used in many different areas in biology and biomedicine, we were wondering how we can make that more useful. Because not everyone knows how to set up an optical structure, right? It's a bit laser physics and etc. required to understand what's going on. So we wanted to make this as simple and as high throughput as possible. And we did that in the form of a new technique that we call real-time deformability cytometry. So this has very little to do with optics at this point, except for imaging. And we've transferred all the hard work to hydrodynamic forces. So what we do is we have cells in suspension in a syringe. We push them through a microfluidic chip with a narrow channel. And the cell is moving at about 10 centimeters per second in a channel that's about 20 by 20 microns, so slightly larger than the cell. And because of the high speed, the hydrodynamic forces that are acting on the cell, so shear forces, shear stresses, and pressure gradients, etc., are sufficient to deform the cell. To capture that, we need to use a camera that's relatively fast, a few thousand frames per second, and then use stroboscopic lighting to freeze the motion of the cells synced with the camera. So you can actually capture how an initially spherical cell enters the channel, elongates, and eventually reaches this bullet-like steady state shape. And we call this real-time deformability cytometry because we're analyzing the images that are coming from the camera in real time. Right? So when a picture comes to the computer, we know whether there's a cell in that image, what the contour is, what you see in red here. We can then calculate something like the cross-sectional area as the proxy for the cell size. We can calculate the circularity of the object. So the further away from a circle it deforms, the larger the smaller the circularity. And then 1 minus that is the deformation. So for every single cell that passes through, you can then calculate cell size versus deformation. And that's what you see in that scatterplot. So every single point there is a single cell measured. So that means we can now continuously measure mechanical properties of cells at rates of 100 to 1,000 cells per second, right? And I want to contrast 100 to 1,000 cells per second with this new technique RTDC to the optical stretcher that we were using previously. So with this optical stretcher, you see a cell as it's flowing down that microfluidic channel, and then you trap the cell with light, which you don't see here. But then there's a certain deformation. You do the measurement, you let it go, you measure the next one. And that's at a rate of about 100 cells in an hour or something like that, maybe a little bit faster. But contrast that to 1,000 cells per second. Right, so you see a screenshot of these cells whizzing by, and you see how quickly this scatter plot here populates. There's a cell counter running at the bottom. So that's maybe not quite 1,000 cells per second, but a few hundred for sure. And this is just so amazingly fast, right? This is 36,000 times faster. Or put differently, we can now measure in a single day as many cells as with the optical stretcher in 100 years. This is this has changed completely what we're doing. This is truly disruptive technology. So nobody really wants to work with this optical stretcher in my group anymore, and everyone is excited about this new technique. And as a consequence, we've had a lot of projects that we could suddenly tackle with all kinds of collaborators very quickly, generate really interesting and new data, new insight. And so you see, I don't know, just a quick snapshot of a couple of papers that we've had in recent years. And I want to focus on one that I think has particularly far-reaching consequences, and that is that we can now take a drop of blood, just prick a finger, right, a few 10 microliters of blood, dilute it, send it through the chip, and measure the mechanical fingerprint of blood in a person. And in this snapshot, you see how the cell suspension actually comes in from the right and then widens to this heart shape. There's some clear sheath fluid that kind of helps to focus the cells into that narrow channel on the left. And then at the end of that channel, this is where we do our measurement. And this is a movie, so I want to let this run just to give you a sense of the speed with which we can analyze cells now. We need that speed because only 1 in 1,000 cells here is actually not a red blood cell. Red blood cells are very dominant in blood, and then you have a few immune cells that we're actually interested in, as you will see. So how does the mechanical fingerprint of blood look like? This is a typical plot here. So you see all these red blood cells, very huge population, outnumbering all the other cells by far, and they're also much softer than the other cells. So they deform quite a bit and move very far up in that scatterplot. And then you see that there are also other populations, other cells, thrombocytes, which make the blood clotting, lymphocytes, and neutrophils is one of the myeloid cell types. So now we can take a drop of blood, run it through the machine, and count how many cells there are of each kind. This is what a doctor does with your blood in when it takes blood. Call it complete blood counts. We can count how many cells there are of each kind. But we can do more than that. We have also information about the size and the deformation of every single cell. And there is additional information there, as I've been telling you. So we've now tried to use this to diagnose certain diseases or detect certain disease conditions. For example, if you take blood from a person with a bacterial infection, different kinds, and then you look at the means of the populations of the lymphocytes and the neutrophils, you see that for a normal person, a healthy donor, these are the gray dots in these plots here. So they have a certain distribution, and the circles, the ellipses here, these are 1 and 2 standard deviations of a normal distribution that you find in a healthy person. And then you can compare that to what you find in a diseased person. So the lymphocytes, they kind of scatter all over. They can be larger, they can be smaller, a little bit more or less deformed than they should be. There's no real clear signature. Many of them are well within the norm. But the neutrophils are different. The neutrophils in these bacterial infection patients are all larger and all more deformed. Right? So that seems to be a signature of an infection in a patient. But a viral infection looks different. Here, the lymphocytes seem to show a very clear response to larger and more deformed, and the neutrophils don't do much. Right? So that there seems to be even difference in the signature between a bacterial and a viral infection. And you can imagine that in a day and age of a global pandemic, where we are wondering how to detect viral infections quickly and safely and efficiently, sensitively, that this might be an attractive alternative to what's currently being done, looking for the viral DNA, to take a drop of blood from a patient and just figure out whether there's actually a viral infection going on or not, and not just a bacterial infection, which would require different treatment, right? So that you can then probably also only then follow up specific molecular level diagnosis for the patients that actually have a viral infection. So we also have been measuring COVID-19 patient blood in Erlangen, but unfortunately, or fortunately rather, there were not very many cases, so we don't have any good numbers yet. Anyway, so this gives you an idea of what we can and will do with this technology. 2 of these machines are already with our collaborators in the hospital, and we're measuring patient blood on a routine basis to figure out what we can really diagnose with that technique. We cannot just analyze, we can also sort, because our analysis is done in real time. We know that information fast enough that we can couple that to sorting using interdigitated electrodes on a piezoelectric substrate to set up surface acoustic waves, then can specifically kick off cells of interest into a target outlet, while all the other cells go to a default outlet. So in addition to imaging and image analysis, we've also added fluorescence. So this is like a normal flow-based, fluorescence-based flow cytometer. And just to show you that we can, if you have a population of 2 different kinds of objects, these are beads with the same area, same size, but different fluorescence level, we can sort for, say, the ones that have high fluorescence. That's what you do with FACS, or fluorescent flow cytometer normally. But what no other technique can do at the moment is to sort for mechanical properties. Right, so this is now a plot of, again, deformation versus area. And you see 2 populations that have about the same size area, but different deformations. So the more deformed ones are softer and the others are stiffer. And we can now say, well, sort for the stiff objects. And this is something that has never been shown before, and opens up a lot of new interesting research possibilities. Before I get to the mechanical part of that story, I want to show you that this also works for cell populations. So if you take blood, again, you have these 3 main cell types: red blood cells, myeloid cells, which contain neutrophils, and the lymphocytes. And we can sort for either one of them just based on the images that we have. And so far, I told you about size and deformation, but obviously there's a lot more information in an image. Right, so this is just a couple of cells that we see sometimes in our RTDC when they pass through. And if an image says more than 1,000 words, then you can imagine how much information we're gathering. If you can measure 1,000 cells per image per second, you get 1,000 images per second, right? We've amassed over the years now, I don't know, something like a few 100 million cell images with specific information about what they are and what disease they stand for, etc. So that's a perfect training ground for artificial intelligence algorithms, which is something that we've now been pursuing actually for some time. So I also want to show you something that we can do now, because if you send your cells through and you label specific cells of interest with an antibody, and fluorescent antibody, so we can detect which cells they are. We train your algorithm to identify the cells based on the images only, and go through some iterations of the network to optimize parameters, so detection accuracy versus inference time. And this should work very well in principle, and it's also what we can do now, so that once you have trained your algorithm with the fluorescent labels, you can then let go of the fluorescent label and only sort the cells based on the images. And that works with very good sensitivity and specificity. So in a sense, we can now transfer the molecular specificity of an antibody-based fluorescence label to label-free sorting, which is something that, again, opens up a number of really interesting avenues to pursue, in particular also in sorting cells for later transplantation into humans or patients, because you cannot have antibodies in those samples. But I want to come back to the mechanical sorting, because something that we're also doing is now that even if you have a single population, there's a distribution of the mechanics, right? So that some cells are more deformed, others are less deformed, and we can sort for the softer cells or the stiffer cells, separate them, and then do a subsequent omics type analysis where we get at transcriptomics or proteomics to identify what is unique to the cells that are stiff compared to the ones that are soft. Right, so you want to figure out what makes a cell soft and what makes a cell stiff. And if we know that, we hope that we can then arbitrarily make cells softer or stiffer. And if you think of mechanics, for example, in the context of circulation, a stiff cell will likely get stuck at small parts of the capillaries. So if you make the cells softer, they might circulate better, or a cancer cell that spreads is softer, maybe you can make them stiffer and prevent them from spreading and metastasizing to other parts of the body. So these are only some of the ideas that we have. I think in general, the control of cell mechanics, so again, physical properties at the cell scale, is an important next step in this field, going from this molecular level control to effects on the cell scale. Now, I want to switch gears a little bit, because so far I've been talking about properties of the cell as a whole, right? The optical structure pulls on the outside, this RTDC deforms the cell from the outside, and this is just the entire object. But can we also reach in and feel inside of cells? Right? Can we assign different mechanical properties to different parts of the cell? This is actually quite exciting area of research. And we had this really great idea that we could use the optical structure to do that, to, for example, measure the stiffness of the nucleus inside of a living cell. Because you get transfer of momentum light to an object at interfaces of refractive index. And the most obvious refractive index jump occurs from the surrounding medium, which is water, usually 1.33 refractive index, to the cell. And that's 1.37 or something like that. But the nucleus is a very dense object inside of the cell. So everyone was like, okay, well, it has a higher refractive index. We're probably also exerting forces on the nucleus. And in order to bring that out very clearly, we had this idea that we could index match the surrounding to the cytoplasm of the cell. So bring that up to 1.37 in order to have the nucleus stand out, and then apply stress on the nucleus only, and then you can deform the nucleus inside of a living cell. That was a great and marvelous idea, but it never worked. We couldn't even trap them. So once we index matched the cells, we could not trap the cells anymore. And why could that be? If the nucleus has a high refractive index, we should be able to do that, but we didn't. So that means the nucleus probably doesn't have a higher refractive index, which was very inconceivable at the time. But then we set out to measure that directly in a number of different ways, and one of which involved another variant of this optical structure. So in this case, we call this an optical cell rotator, where again you have a two-beam trap, but we don't use two single mode beams. We have one of them with a higher order mode. And we excite that higher order mode dynamically with a spatial light modulator. So we can rotate the mode in the fiber. And because the modes will then address particular refractive index heterogeneities in the cell, we can also rotate the cell with these modes. And because this optical fiber, the optical trap, this dual beam trap, is not coupled to the imaging optics, like with optical tweezers typically, we can actually rotate the object through the imaging plane of that microscope. And if you do that with a microscope that can calculate or capture the phase information quantitatively, so we do that with a digital holographic microscope, we have a reference beam and an object beam, interfere that on a camera, and then you can calculate the phase from the fringe pattern that you get. If you do that on cells from all kinds of different sites, you're capturing two-dimensional quantitative phase information from all kinds of angles. And then you can use tomographic reconstruction to really figure out where in the cell is what kind of refractive index, right? So if we do that, we really find that the area in the cell that we can, for example, also stain with fluorescence, so we know where the nucleus is, indeed has a lower refractive index than the rest of the cell. So this is the white dashed line there. The yellow, very high refractive index blob is the so-called nucleolus within the cell nucleus, but that's too small to be relevant. And the reason why we could never trap the cells in this optical trap when we indexed matched the surrounding was because the nucleus had a lower refractive index, and that is not stable in a trap because the gradient forces revert and reverse, and so the objects are pushed out of the trap. We cannot trap that. And even when you step through, you see this dark void here with lower refractive index. That's the nucleus. We also confirmed that by now on attached cells. There was some suspicion that maybe the fact that the cells are suspended changes properties of the cell nucleus, but that's not the case. Also in attached cells, we see that the nucleus has a lower refractive index. Here we don't rotate the cell, obviously, but we use a galvanometer to illuminate the cell from different angles. Quite surprising, nobody expected that, but I think this is now very clearly confirmed by us and also others that the nucleus has lower refractive index and density, and also that has consequences for how you can image into cells and tissues, etc. But this didn't work. So this idea to measure, use the optical stretcher to measure nuclear mechanics or anything inside of the cell. But this ODT, this optical diffraction tomography, is actually quite useful in another combination of yet another optical technique, and that is Brillant microscopy. Now, I don't need to tell you what Brillant scattering is. If you have any questions, actually ask Birgit, who's speaking right after me. But essentially, that's the inelastic scattering of photons by acoustic phonons. So the Brillant shift that you're getting in the spectrum is directly related to the longitudinal modulus of the material, right? It's also proportional to refractive index, and the density inversely proportional to the density. So that's something that is important to consider. But wavelength and the angle under which you detect is fixed. So in a sense, with the Brillant shift, you can do measurements of elasticity inside of an object, right? Light goes into material, so you look at the light that comes back, and you can get information about the material on the inside. Now, the ability to measure the very small amount of photons that is coming back from this process and quantitate the spectrum has shifted significantly. This was something that was done by tandem scanning Fabry-Perot for the longest time, which is very sensitive, but it takes a long time until you get through your spectrum. And if you use bypass, which were introduced by Giuliano Scarselli, you can actually speed that up to the point where a single measurement doesn't take minutes, but a fraction of a second. So now you can do a confocal measurement of properties of the Brillant shift inside of cells, and you build up three-dimensional information of the distribution of this longitudinal modulus, or at least the Brillant shift, inside of a cell. Now we've done that too. We have a few of those setups now in our labs. And we've used that, for example, to look into living zebrafish larvae. We're interested in them because they can repair their spinal cord after injury. And I said that we're interested in mechanosensing in the central nervous system. So that's kind of part of that research to figure out what are the mechanical properties of the spinal cord, the brain, the retina in a living animal. And here you can see that we can actually get orthogonal planes of the distribution of the mechanical properties of the Brillant shift in a living larva. Right? I could tell you a lot more about that, but I don't have the time to go into details here. But I want to point out one aspect, and that is if you want to measure really the longitudinal modulus, you need to know the refractive index and the density. So that's why we have now combined Brillant microscope with this optical diffraction tomography setup, and also thrown in fluorescence for good measure, which means that we can now, for example, in a single cell, identify objects of interest by fluorescence. We can measure the refractive index, calculate the density from the refractive index, and then get proper information about the longitudinal modulus. So with that, we are now at the point where we can measure mechanical properties of organelles inside of cells. Really reach in and figure out what is the stiffness of the nucleus or the mitochondria, or in this case, actually, these are phase-separated protein droplets. As I said, this is one of the hot topics in cell biology at the moment. This is where we are at the moment, where do we want to go with this? Ultimately, we would like to have a setup that is working similar to a light sheet microscopy. And I should say thanks to Jan Husken for these beautiful movies of the development of a zebrafish embryo over several tens of hours of embryonic development, where you see the development of the immune system actually in this fish. You see these immune cells moving about. And we are setting this up at the moment, but we want to combine that with, again, Brillant microscopy and optical diffraction tomography, so that we can do mechanical measurements inside of living organisms, right? The idea being that we want to measure the stiffness of the cells as they move about. Are the ones that are particularly fast also particularly soft? Can we stiffen the tissue? Can we impact what is going on inside of this fish? And now I think we're getting close to having the right tools to do that even at an organismal animal scale with subcellular resolution, but not looking for structure, really measuring physical properties. Now with that, I come to the end of my presentation. I hope that you are seeing that what I wanted to say at the beginning, that light can do more than just imaging structure, that you can actually measure physical properties. And there's also more to optical trapping than optical tweezers. We've had a lot of fun with these dual beam laser traps, which are now being replaced by other tools such as Brillant microscopy and optical diffraction tomography, because they can actually get information about really highly resourced spatial information inside living cells and animals. And I also hope that you see why I say that physical properties of cells and tissues are important to understand biology and have the potential to impact medicine. This is my team, great bunch of people. This is our funding. We are on Twitter if you want to follow us in our research. And I thank you very much for your attention, and I'm looking forward to your questions.
Everybody's clapping, but regrettably, you could probably only hear me do it.
Yeah.
So Professor Guck, thank you very much. That was a fascinating talk. It's inspirational. It says that physicists, if they try hard enough, can actually do some biology or biomedicine. I forgot to say earlier that if you have questions for the speaker, just use the question and answer feature at the bottom of your computer screen. But we do have some questions already.
I can go ahead and read them if you would like, Bob.
Yeah, please do, please do.
Okay, so thank you very much, Professor Guck. I'll start with a question from Dr. Zahiro Alam. He says, I can't help to notice that those guiding structures, the ones you had when you're showing the guiding structures within the retina of the eye, that they have a very peculiar shape compared to man-made fibers. Do we know why those structures might help? Secondly, you would think that by letting only a few modes of light through the guiding structure, the eye is essentially throwing away information that is coming to the eye as a sort of mode filtering. Is there an evolutionary or functional advantage for doing so?
Yeah, very good question. So I guess you might have been referring to these broadening structures at the top. These are called the so-called end feet plural. Maybe you see that here. So it's actually interesting if you look at the refractive index in the image at the middle here. The refractive index of the end foot is lower than in the rest of the cell, but the width, the diameter increases. So what that means is that larger diameter but lower refractive index means that the light guiding properties stay about the same. You can see that in the V parameter on the right side for two different wavelengths, blue and red. So they don't differ too much along the length of the fiber.
Professor Guck, sorry, you're not currently sharing your screen. So if you're wanting to refer to elements that are on your slides, could you maybe share your screen again? Yeah, I mean, you're answering the question, but just in case you wanted to refer to your images.
Yeah, I think that would be useful in this case. Thanks for pointing that out. So this is what I was referring to. Can you see that now?
Yes, I can. Thank you.
Yeah, so this is, I guess this were the structures that you were talking about, this end foot, this widening of the cell at the inner retinal surface, close to the vitreous, that narrows towards the bottom. The bottom all the way down, these are the rod outer segments are actually already the photoreceptors, so they're still attached here. The end foot has a lower refractive index than the rest of the cell, but it's wider. So the waveguiding properties stay about the same, but the refractive index is lower. So we think that this might be helping to, number one, it's wider, so it covers a larger area, but it also has lower reflection because the refractive index is closer to the vitreous, to the refractive index of the vitreous. So you probably get more light into the cell, and then you can condense it down to a thinner diameter by increasing the refractive index, and then creating a lot more space for the other cells that are still there. The soma, so the cell body, is kind of sitting on the outside of these two processes. It's not really in a way. There are very few mitochondria, smaller, larger, typically rather scattering structures that you don't find in these cells. So I think, yes, so the particular shape of the cells has been optimized to get light through. But the second question was then whether the cell is throwing away information because it only guides a few modes. So I think there is not much spatial information that needs to be conveyed because there's a one-to-one pairing of the size of the end foot. So one Müller cell goes down typically to one cone photoreceptor. So this is the spatial information that you can sample anyway. You're not losing information in that sense. I think it's more important at low light vision to make sure that all the photons that are actually hitting the retina make it all the way down to the level of the photoreceptors, in particular to the rods at night, to be detected.
Thank you. The next question I have is from Professor Ebrahim Karimi. In addition to complimenting you on an excellent talk, he's interested to know about how the intensity of light, depending on the wavelength, will affect any cell mechanism. And with that in mind, could you consider implementing some kind of quantum illumination technique, i.e. something that's interaction-free or uses computational ghost imaging, in order to minimize any such effects? This maybe be an extreme end of what could be called low-dose illumination.
Right, so excellent question. Of course, it matters what wavelength you use because the cell contains chromophores, so things that the cell absorbs. In particular, as you start in the UV, there's still a lot of absorption, which kind of drops out towards the longer wavelength. So red is okay, near-infrared is better. That's why we typically use some near-infrared laser beams for really hitting the cell hard with, for deforming it, for example. But as you go further into the near-infrared, you know that the water absorption increases again. So that's an optimum somewhere between, say, 750 and maybe all the way out to 1060 nanometers, which are typical wavelengths to be used. So it definitely makes sense to also think about other ways, other schemes to reduce the number of photons that are going through. And yeah, using quantum effects or ghost imaging are really clever ways of implementing that. I think there's certainly an area of application for these kind of approaches, yes.
Okay, I think I have time for one more question. And any questions that remain, I will send to you so you can answer them directly with the person if need be. My next question is from Professor Arno Weck. He asks, is there a way to change the stress state that's applied on the cells, say tensile or biaxial tension, shear, etc., by using multiple beams? Could some stress states be more revealing of the cells themselves?
Yeah, I think it's a good question. So the cells certainly have anisotropic properties, both in their mechanics, but also in their optical properties. And so it could be interesting to test from different sides. The problem is that with the, say you have two laser beams and they have Gaussian modes, then you have one axis and the cell will always orient along that axis with the major anisotropy, usually the shape, or if there's a strong optical anisotropy, then this will be defining the axis along the cell lines, and then you can mostly stretch it along that, right? If you then have a second pair of lasers that come in, you might be able to do a little bit of something, but as soon as this becomes stronger than the trapping beam, the cell will just flip. So we've never figured out a way to test other modes than the most dominant one, biaxial stretching. I don't think we know how to do that with light, but maybe it's just something for to try by somebody that's smarter than us.
Okay, thank you. And as I said, I will send you the remaining questions separately with the information of the individuals in case you want to answer questions that we couldn't make time for. Thank you once again for an excellent talk. I'm happy to return control to Bob.
Thank you very much. Bye.
So, Jared, thank you very much for handling the question and answers. And I totally agree. We give our great thanks to Professor Guck for this fascinating talk that he just gave.